Hello everyone. Today I will be discussing the paper of MBBS first professional examination on the subject of biochemistry. Now the first question is: a middle-aged woman rushed to emergency with pain, abdomen and nausea. On asking the patient, confirmed dark urine and grey stool. Point the mark here: dark urine and grey stool. After managing the emergency situation, blood samples were tested for liver function. Results are given as total bilirubin 6.2 milligram per deciliter, direct bilirubin, that is conjugated bilirubin, is 5.8 milligram per deciliter. Total protein is 7.2, albumin is 3.8, AST is 26 in normal range, ALT is 32 usually in normal range, alkaline phosphatase is 387, that is much more in range, and GGT is 35 units per liter. Okay, so what are the questions? The questions are: as an attending medical officer, what would your provisional diagnosis? So I am giving the answers here. The provisional diagnosis for the first question is obstructive jaundice. Why obstructive jaundice? As here I can see that the direct bilirubin is much more as compared to indirect bilirubin or unconjugated bilirubin. How should I calculate here? 6.2 minus 5.8 is remaining 0.4. This is the unconjugated bilirubin. So this means unconjugated bilirubin generally increases in hepatocellular, I would say, and hemolytic jaundice. So if I consider just the obstructive jaundice, the direct bilirubin is much more level. Also here alkaline phosphatase is much more. You know that alkaline phosphatase is a surface enzyme, and surface enzyme generally increases in outer damage of the liver, means not intracellular damage of the liver. So outer cellular damage generally occurs in the obstructive jaundice, and if it happens hepatocellular, then alkaline phosphatase along with AST and ALT will be high, but here only alkaline phosphatase is high. That means I am saying that it is obstructive jaundice. So the answer is obstructive jaundice. Now the second part is: what all other tests would you suggest to justify your diagnosis? In this question here it is already given that liver function test is done. So the other tests which will be said are the LFT is already given, CT scan, MRCP means magnetic resonance cholangiopancreatopathy, and ERCP, endoscopic resonance cholangiopancreatopathy. Here a tube is inserted in the lumen of the bile duct and seen whether there is any kind of obstruction or not. Okay, this obstruction is removed later. For which type of obstruction is seen according to it. Okay, and endoscopic ultrasound can be done. So these are some of the tests that can be done for the checking of whether there is obstruction or not. But here you can see that CT scan, MRCP are relatively higher in costing, so LFT is much more cost effective than the other ones. Now the third one is: how would you exclude acute pancreatitis or alcoholic? So I am giving here an example here: pancreatitis and alcoholic obstructive jaundice. In both the cases, here in pancreatitis and alcoholic, in both the cases obstructive jaundice occurs. Now here in pancreatitis there is inflammation of pancreas, okay, and in alcoholism there is chronic consumption of the alcohol. How it affects the bile duct? Now the pancreas here generally present, sorry, in the image I have done a little bit mistake. Damage will be here and another damage will be here. Okay, so if I see the alcoholic one, alcohol direct damages the liver. Okay, so if the liver is damaged or it gets fibrosed, means scar tissue formation, the right hepatic duct and the left hepatic duct will be obstructed, thus the flow will be stopped and there will be obstructive jaundice. But in pancreatitis, if there is inflammation in the pancreas, the both duct from gallbladder and the liver, that is a common hepatic duct, along with the pancreatic duct, will be obstructed. So overall the flow of the bile duct is hampered. Now, other points include, in pancreatitis there is abdominal pain, nausea, vomiting, jaundice, etc. And in hepatic, or I would say alcoholic obstructive jaundice, there will be fatigue, weight loss, ascites, means fluid filled cavity in the abdomen. In both the cases, there will be case presentation of dark urine and pale stool. In the question, it is already given grayish stool and dark urine is passed by the patient. And how would I divide this much more, or I should say discriminate between these two? While testing the patient, we would see the pancreatic lipase or amylase is high or not. If the pancreatic lipase or amylase is high, there is higher chance of pancreatitis. And LFT along with dark urine and pale stool just indicates the alcoholic liver disease, or I would say alcoholic obstructive jaundice. Along with that, the history of the patient is also taken. Now the last one is, what are the different causes of conjugated hyperbilirubinemia? Causes. The causes are, first main cause is the bile duct obstruction. Bile duct obstruction can be done by the formation of tumor at that bile duct site, or there can be gallbladder stone which gets stuck up in the GB duct or the common hepatic duct, or there can be any other inflammation due to some kind of infection or the pancreatitis, etc. The other points include the liver disease. There can be hepatitis, cirrhosis, etc. In cirrhosis, there will be formation of the scar tissue and fibrosis, which will obviously obstruct the hepatic duct. And there are some kind of medications like ciprofloxacin, TCA, tricyclic antidepressants. The tricyclic antidepressants acts as the antidepressing medicine, and halothane. Okay. And other points include the pancreatitis or any kind of sepsis. Now the second question is, a patient with chronic renal failure is admitted in comatose state in hospital. Here the point is comatose state in hospital. On clinical examination, features of the encephalopathy are seen, and blood investigation shows creatinine level of the patient is 6.5 milligram per deciliter, which is high. Okay. And urea level of 425 is definitely much more higher. So first question, what describe the metabolic dearrangement responsible for this condition? The metabolic dearrangement here is hyperammonemia. Now hyperammonemia occurs. First of all, we should remember the urea cycle. Here the wasted products are the ammonia, and the main point here given is the carboxyl phosphate synthase. Sorry, the point is little bit okay. The main waste product is ammonia, and the ammonia is changed to carboxyl phosphate, and it enters in the urea cycle. The main transformation is done by the carboxyl phosphate synthase 1. This is the rate limiting enzyme in this whole process or the whole metabolism. Now the next question is, explain the biochemical basis of ammonia toxicity in the brain. The ammonia toxicity affects the brain in many ways. First, there will be a glutamate enterotoxicity, and in glutamate toxicity, the NMDA receptors are damaged, and the calcium influx occurs in huge amounts. The calcium influx leads to hyperexcitability of the neurons, and other nerve tissues which can be deleterious for the brain. Now the second is the alpha ketoglutarate dehydrogenase inhibition. This is the main enzyme for the glucogenesis or the glycolysis process, and if there is inhibition of this enzyme, no glucose or nor the energy will be formed in the brain, and it will lead to tissue damage. Third is disruption of the formation of the GABA, glutamate and other neurotransmitters. GABA and glutamate are very important neurotransmitters in the brain. If it gets depleted, it will definitely lead to some kind of alterations in the brain action. Fourth is inflammatory response. If ammonia gets accumulated in high amount, it will change the osmolarity of the tissue and there can be inflammatory response. Now mitochondrial disturbed function. If mitochondria gets disturbed or it has altered function, the ATP production will be decreased, and if ATP production is decreased, it will definitely lead to decreased brain activity or decreased brain functionality proper. Next is oxidative stress. Oxidative stress means reactive oxygen species is increased in the brain tissue due to the accumulation of ammonia or ammonium ions. And finally is the blood brain barrier damage. If blood brain barrier damage, there will be infection, there can be other deleterious things which may be affected on the brain. So these are some of the points which are affected due to ammonia toxicity. Now the third question is, I would say, second question, first part. Name the rate limiting enzyme of fatty acid synthesis. Describe the regulation of it. Discuss how this enzyme's activity influences beta oxidation of fatty acid. Name the cell organelle where the fatty acids can be desaturated and elongated. Big question, huh? So first one, the rate limiting enzyme is the acetyl CoA carboxylase. Okay, this is the main rate limiting enzyme. Now the regulation of the enzyme or the whole process can be done by mainly three processes. One is the allosteric regulation, one is the transcriptional regulation. Second, mark it a little bit. First is allosteric regulation. As the name suggests, allosteric means in the different site of the enzyme, and there it is attacked, and there will be regulation of whether the enzyme production will be more or whether the enzyme production will be less. Transcriptional regulation. When the DNA to RNA formation is altered, there will be transcriptional regulation, as the name suggesting. And the last one is phosphorylation or the dephosphorylation. Majority of the enzymes act by the process of phosphorylation or the dephosphorylation. So if there will be phosphorylation or the dephosphorylation, the enzyme will be activated or inactivated respectively. Now the third part is the discuss how this enzyme describe the regulation of it, already described. Discuss how this enzyme's activity influences beta oxidation of the fatty acid, and name the cell organelle where the fatty acids can be desaturated. First I should answer the cell organelle. The cell organelle is endoplasmic reticulum. Especially in the smooth endoplasmic reticulum, here the desaturation or the elongation of the fatty acids is mainly done. And now the beta oxidation of fatty acid. Beta oxidation of the fatty acid is controlled by the enzyme acetyl CoA carboxylase. Regulates the activity of malonyl CoA. Malonyl CoA inhibits the CPD1. CPD1 is carnitine palmitoyl transferase 1. As you should remember, it is helpful in the transportation process of the fatty acid from the mitochondria to cytosol and vice versa. Okay. CPD1 is much required If it is regulated, then overall the availability of the malonyl CoA will be regulated. Now, ACC or the malonyl CoA favors the fatty acid synthesis, not on the beta oxidation, and high level of malonyl CoA reduces the beta oxidation process in the cell. So these are some of the regulation process of, I would say, acetyl CoA carboxylase in the beta oxidation of the fatty acid. And answer here is the endoplasmic reticulum. Now the next question is, describe the process used to separate cell organelles and release the marker enzymes of cell membrane and subcellular organelles. The process is called the cell fractionation. Cell fractionation, or it is also called differential centrifugation. Here the main steps are the homogenization. In homogenization, the cell membrane is broken down and homogeneous mixture of the cell is formed. Not like that the cell membrane is intact and in it the cytosol is present, nucleus and other cell organelles are present. They are disintegrated at all. Okay. After that, the next process is the centrifugation. The centrifugation can be done in four processes, I would say. 4 processes or 4 levels. Low grade centrifugation, medium grade centrifugation, high grade centrifugation, and finally the ultra centrifugation. And these are the main processes by which the cell organelles are divided. And other processes include, which are also done for the differentiation or the process of differentiating the cell organelles, is the electrophoresis or gel filtration. Now, the marker enzymes for the cell organelles. The main organelles are, the first of all is the cell membrane. So at cell membrane, as I said earlier, the ALP is present. ALP, what is ALP? Alkaline phosphatase. It is a surface enzyme. ALP can be a marker. The sodium potassium ATPase enzyme can be a marker. In endoplasmic reticulum, mainly the cytochrome 450 is present and glucose 6 phosphatase is present. So this can be act as marker. In mitochondria, there can be malate dehydrogenase cytochrome C oxidase. The main enzymes I am marking here: sodium potassium ATPase, cytochrome 450. In mitochondria, it is cytochrome C oxidase. In cytosol, there can be pyruvate kinase. These enzymes you should remember. In Golgi apparatus, there can be galactosyl transferase, as the name given here, G and here G. Just remember this mnemonics. And in lysozymes, it can be phosphatase or the beta galactosidase, and in peroxidase, the catalase is the main enzyme. Now the third question. A 55-year-old female is complaining of joint pain. Her autoantibody profile is within normal limit. Her autoantibody profile is within normal limit means it is not any kind of autoimmune disease. In autoimmune disease, there can be rheumatoid arthritis present, and in which joint pain is a common feature. So here the profile is normal means we can rule out the rheumatoid arthritis. So it is probably the case of gout, let's say. The first question is, which biochemical investigation should you advise for the patient? Write the physiological reference range of that parameter. Describe the metabolism of that compound and give a short note on the management of this patient. The answer is gout. Now we will check the patient's blood uric acid concentration level and synovial fluid examination. In synovial fluid examination, there will be presence of urate crystals or uric acid crystals. If the uric acid crystals is much more level, then it is a case of gout. And generally in healthy persons, the blood level for blood uric acid concentration is 3 to 7.5 milligram per deciliter. I should mark these points: blood uric acid concentration, synovial fluid examination, and the normal range is 3 to 7.5 milligram per deciliter. Now the metabolism of uric acid. Should transfer it here. Okay. Now the metabolism. In metabolism there is breakdown of AMP and GMP. What is AMP? You should know this answer, and GMP you also know this. From the AMP the adenosine is abstracted, and from GMP the guanosine is abstracted. GMP means guanosine monophosphate, and guanosine to guanine and adenosine to inosine. Okay. Now using these enzymes it is reduced to finally uric acid, okay, xanthine, or there can be another one, that is hypoxanthine. Okay. Now the treatment. In the treatment procedure there can be many medicines present. First medicine can be NSAIDs, means non-steroidal anti-inflammatory drugs, corticosteroids, steroidal drugs, and other advices will be weight loss. If the patient is healthy or too much healthy, I would say, the weight loss is a required point, and less alcohol intake, and the patient has to be advised that he or she should take avoid purine rich food. Purine rich food means eggs, red meat, etc. Okay. And other medicines include the allopurinol. Allopurinol is negatively acting on the xanthine oxidase enzyme. If it acts on the xanthine oxidase, the uric acid formation will be halted from xanthine, and xanthine will be easily dispersed from the body. Now the next question is attitude of doctors to patient and patient parties. This is an AdCom question. I should not explain this question, you should write it on your own. I hope you have written this question very easily. The next question is transamination reaction. So what is transamination reaction? Transamination reaction is transfer of amino group from one amino acid to keto acid and new amino acid forms. Means from one amino acid with keto acid forms amino acid 2 from this one only, and new keto acid from this amino acid by the transferring of the amino group. Okay, amino group means NH2 group. So this transamination is very much required because it is required for the metabolism of nitrogen, new amino acid formation. There are many non-essential amino acids in our body. How they are formed? They are transformed from one amino acid to another amino acid by the transamination processes. Okay, so this is a process of new amino acid formation. This reaction occurs by the transaminase enzyme. Okay, and this reaction is reversible in nature. Means the point I should make here is like something like this. Sorry. means it is reversible in nature. Some examples include, you know this example already, that is glutamate to glutamate and alpha ketoglutarate forms the aspartate and glutamine. Next question is, explain the following statements. Histidine has an important role in buffering action of proteins. So, histidine has a pKa value of near 7. Near 7 means it is neutral in nature, and if it is neutral, then it can both accept H plus and donate H plus, means proton accept and donate, and this property can stabilize the pH sensitive functional groups. If pH sensitive functional groups are not saved in harsh pH conditions, the action will not occur. So it can stabilize the pH sensitive functional groups. It can accept H plus, it can donate proton, pH sensitive functional groups. It can regulate the enzyme activities at different pH or different harsh environment. Helps in the redox reaction, as both positive and negative charge can be controlled, so it can take place in the redox reaction, and it gives protection from the oxidative damage. So these are the some points which if you could write in the explain why, you could get full marks. Now the next question is, creatine clearance is important for the checking of renal function. So what is creatine clearance rate? Creatinine clearance rate is to check the kidney's ability to filter the creatinine in a per unit time. So it is to check the kidney's ability to filter waste products, as creatinine is also a waste product and excreted from the kidneys. So it can be used as a parameter to check whether the kidneys is acting okay or not. So checking the renal function, renal impairment whether present or not, or any kind of CKD present, chronic kidney disease. If there is any medicine taking by the patient and it leading to kidney damage or kidney disorders or alteration in the kidney clearance rate, that means the medicine has some toxicity on the kidney and overall body. So to alternate the medicine dosing or alternate the medicine at all. Next is the fluid management and checking the body toxicity. If any kind of toxic is present in the creatinine clearance rate, I would say in the liver, and it hampers the creatinine clearance rate, means it is affecting the kidney. The LDL is atherogenic in nature. How it is atherogenic in nature? LDL transports the cholesterol to peripheral tissue. Okay, LDL transports the cholesterol from liver to the peripheral tissues for requirements, but it also can act as deleterious effects. So LDL can penetrate the endothelium of the vessels. Endothelium of the vessels, and in the endothelium it can get oxidized and formation of the foam cells along with the macrophages. Foam cells is formed by LDL oxidized one with macrophages. Okay, foam cell form, and it leads to formation of the plaque formation. What is plaque? Plaque means if this is a blood vessel and these are the lumen, here a plaque something like this forms, and it generally outgrows till it is blocked completely. Okay, this is known as a plaque. and LDL when glycosylated or oxidized, it is much more problematic for the vessels or the CVS. Okay, so it has to be checked at all. Now the next question is on defects in G protein mediated pathways may lead to disease like cholera. So in cholera there is cholera toxin present. So how the cholera toxin affects the G protein, let's see. The cholera toxin binds to the intestinal epithelium. Okay, binds to intestinal epithelium, and the G proteins are present on the surface of the intestinal epithelium. Okay, so the G proteins get activated. The activated G proteins increase the CAMP concentration, cyclic adenosine monophosphate concentration, and this CAMP concentration increased leads to stimulation of the chlorine release and sodium absorption is decreased. Means so chlorine channels get opened towards the lumen side and more amount of chlorine is released, and the sodium channels are gets blocked. Means the chlorine release is more and sodium absorption from the lumen is reduced, leading to change in osmolarity in lumen, and changing in osmolarity of lumen leading to more water retention in the intestine, and more water retention in the intestine means diarrhea. So the cholera toxin mainly promotes the water loss, and the patient comes to the doctor, or the doctor's that the patient has rice watery stool for last 2-3 days. First there is mild fever and all, and then there is a watery stool. Also the cholera toxin alternates the G protein. How it alternates the G protein and making it more sensitive, and if it gets much more sensitive in lower concentration of G protein activators and the cholera toxin, the G protein gets easily activated and the whole process gets take place, and more diarrhea will occur, and more water loss and iron loss will occur. So the main treatment includes the ORS treatment, and the bacteria has to be removed. Now next question is Carnicus may occur in type 1 Crigler-Najjar syndrome. Crigler-Najjar syndrome is an enzymatic defect disease, so it occurs mainly in small children. Now what is Carnicus? Carnicus means brain dysfunction due to increased bilirubin brain dysfunction due to increased bilirubin in the brain tissue. The bilirubin is generally unconjugated in nature. And in Crigler-Najjar syndrome type 1, there is deficiency of glucuronil transferase. This enzyme is defective in this syndrome. Thus, the whole metabolism of the bilirubin is altered, and the unconjugated bilirubin is increased in the child's body, and it gets accumulated in different tissues including the brain, as it can cross the blood-brain barrier and accumulates in brain like the basal ganglia, midbrain, and it affects the children in many ways like loss of consciousness, lower thinking capacity, or lower I don't say IQ, I would say cognitive characteristics of the child is reduced. Okay, and it can be treated with phototherapy or the medicines required for the betterment of the liver, or I would say the glucuronil transferase has to be replaced. It is generally a genetic defect, so it can't be treated easily. Bilirubin cross the blood-brain barrier when the concentration is high. Now the MCQs. First question: methotrexate, a widely used anti-cancer drug, acts by inhibiting. The answer is dihydrofolate reductase. Methotrexate affects the dihydrofolate reductase, thus the folate metabolism is altered, and the folate or the folic acid is much more required for the cell formation. If it is reduced, then the cancer cells will die off. The second question: 1-year-old child brought to the pediatric OPD. Mother complained that child has mousy odor in urine. Mousy odor, remember this point. On examination, it was seen that child may have many delayed milestones and hypopigmentation of hair and skin. Ferric chloride test, important point, ferric chloride test and Guthrie's test were positive. These tests are positive mainly for the phenylketonuria in children. In phenylketonuria, there is defect in the enzyme or decreased enzyme, that is phenylalanine hydroxylase. So this is the answer. Now the next question is: enzymes of glycogen metabolism are under allosteric regulation. Identify from below which of the following is not an allosteric regulator of the same. Insulin is the answer. Insulin is not an allosteric regulator. Next question: end products of one cycle of beta oxidation of odd chain fatty acids are. The answer is acetyl CoA and propionyl CoA. Now the next question is which of the following has pseudouridine in nature? What is pseudouridine? Pseudouridine is uridine with C5 and C1 glycosidic bond present, which is little bit of different structure from the uridine at all, and it can be present in RNA, tRNA, mRNA, all, but in most concentration in tRNA it is present. So out of these four, I would rather suggest the answer will be tRNA. In hospital, used Ryle's tube is discarded in. Ryle's tube, after use, it is infective in nature. So if it is infective in nature, it has to be discarded in yellow bag. Yellow bag is not the answer. In yellow bag, the anatomic wastes are disposed. In white container, the needle syringes are disposed. In blue container, the infective but plastic and glass like things are disposed, and in red bag the infective things are disposed. That means the Ryle's tube is discarded. The precursors for the synthesis of polyamines are. The precursors of the polyamines are the ornithine, arginine and methionine. So here both can be answered, methionine and arginine. Option A and option B both can be answered. Means SAM is also part of the methionine. So both can be answered, but after discussing with the faculties and all, they are saying that the ornithine and SAM is the answer. I don't know, but both can be the correct answer. Next one: the enzyme associated with reverse cholesterol transfer is LCAT. LCAT, the answer question is easy. LCAT is lecithin cholesterol acyltransferase. It helps in the transferring of cholesterol to cholesterol esters. Next question: after being synthesized in reticuloendothelial cells, bilirubin is transported to the liver through which of the following transporters in blood? The answer is simple: albumin. And the last one is 2 years old child presenting with liver enlargement. On investigation, following features were found. Liver enlargement is the main point. In galactosemia, liver enlargement is not seen generally. In fructose deficiency, not seen. In G6PD, it is not seen. So the last answer is von Gerke's disease. So the whole paper is discussed here. Thank you very much.